morning to my teacher and all my fellow friends. As the current situation is going serious, so that I have been forced to present this in my house. Okay, my title of presentation today is Monohybrid Cross. What is Monohybrid Cross? Monohybrid Cross is the cross between two parents involve only one characteristic. Just we can see at here, we use pure tall stem which got tall characteristic, and short stem got only short characteristic with its small capital letter T. And then let them have pollination, so that we can extract their gametes TT and T and small t, and let them have cross section, which results F1, first failure generation, semi-TT. And then after we receive semi-T, we let them have self-pollination again, so that we can get the new generation, which is second failure generation. And we can see this, the result is just only one got pure tall characteristic, and then two of them got semi characteristic, which is got tall and short, and then the last one got pure short characteristic. And then we get results that in the ratio, which is got 3 plants got the tall characteristic, and one pea plant got the short characteristic. This is what we call monohybrid cross. Thank you.
Okay, hi. Today I'm going to present the terms used in genetics. So when we're covering this chapter in biology, we need to know all of these terms. So first we have the gene, which is basically inheritance that determine a particular characteristic in an organism. Example of this is DNA inside a nucleus. Then we have the allele, one of the two alternative forms of a gene that occupy the same locus on the homologous chromosomes. This allele here can be split into two categories: the dominant and the recessive. The dominant is the allele which it expresses itself and displays the dominant trait of the characteristic. And both alleles are dominant, or at least one dominant allele is present. So it means that if both alleles are dominant, or at least one of it is a dominant allele, then it will represent a dominant trait which is known in your biological component. The recessive, then we have the recessive allele, which is the allele that expresses itself in the absence of a dominant allele. So when there is no dominant allele, it will express itself to replace it. After that, we have the phenotype, which is essentially the physical appearance of an organism, which is an observable characteristic. So when you go out and your face is handsome or ugly, and this is the cause. That is the cause, which also differentiates people from people. After that, we have the genotype, which is the genetic composition or the genetic content of an organism. Then we have the homozygote, which is the organism with two identical alleles for a particular characteristic. So basically this right here shows a particular characteristic if two of the alleles are identical. Next we have the heterozygote, which is similar to homozygote, but instead of identical, it's two different. So example, one of it is dominant, the other is recessive, and one of it is dominant, the other is recessive. And then we have different types of allele with different pairings. Lastly, purebred, which is population of organisms which has a particular trait that remain unchanged for many generations. So when you get married, have kids, then some of your kids might retain some of your genetics or some of your wife's genetics. Thank you.
Hey guys, your mate Martin here. Today I'm going to discuss about the blood group A, B, O. I'm going to divide this into two parts. This one is the first and the next one is the second part. So you'll come across this word very frequently in this chapter, genotype. Genotype is actually a genetic, is a genetic makeup of an organism, and allele is an alternate form of gene represented by the letter. Usually it's represented by the letter I. Okay, let's start this. A combination of IA, and IAIO is going to give you a blood group or a phenotype of blood group A. The antigen found in the red blood cell is A and the antibody is anti-B. A combination of IBIO is going to give you a blood group of B, and the antigen is B and the antibody is anti-A. And this is a little special. The genotype IA, the combination of IAIB is going to give you a blood group of AB, is also known as the universal recipient. The antigen is known as A and B, and the antibody form is none. And last but not least, IOIO, the blood group is also known as universal donor, sorry to say, it's a blood group O. And the antigen form is none, and the antibody is anti-A and anti-B. Thank you. Hey guys. So this is the part 2 of the video. So it's the second most important thing, the cross method. So we're going to take a very good example of a male from a blood group A and a female from a blood group B. And we know a blood group A is made up from a combination of IA and IO, and the blood group B is made up from a combination of IB and IO. So this is how the table looks like, and at the end, when we deduce this, you can come to know that the phenotypic ratio for everything is equal. The newborn baby will be able to get blood group AB, or blood group A, B, or O. Everything is equal. So the moral of me is what I'm trying to say.
Have you ever wondered why you are a boy or a girl? In the earlier topic, you have studied that each human somatic cell contains 46 chromosomes. The human females have 2 identical sex chromosomes called X chromosomes. The males have 1 X chromosome and 1 smaller Y chromosome. It is these chromosomes that determine the sex of an individual, whether you are a boy or a girl. During the formation of gametes in the female, the sex chromosomes segregate. All the female eggs produced contain 22 autosomes and 1 X chromosome. The human males produces 2 types of sperms. Sperms with 22 autosomes and 1 X chromosomes, and sperms with 22 autosomes and 1 Y chromosome. As the mother's egg ovum contains only X chromosome, it is therefore the father's sperm that determines the sex of the child. If the sperm carrying an X chromosome fertilizes the ovum, the child is a female. If the sperm carrying a Y chromosome fertilizes an ovum, then the offspring is a male child. And there is an equal chance of the mother having a baby boy or a baby girl. So that's it for today's lesson. Thank you.
How are you? I hope that you find and healthy. And I'm Darshan here. Today I'm going to present about hemophilia and colorblindness itself. Okay, let's go to that. Hemophilia. Hemophilia is a disease that can be transmitted from parents to the offspring. What is offspring? Just let me. Offspring is the exact meaning of generation, like parents, son, like that. Okay, that's what we call offspring. It can be known as generation. Okay. Hemophilia is a hereditary sex-linked disease caused by a recessive allele called on the X chromosome. Okay. First of all, what is hereditary? Hereditary is a generational disease. Like, let me give you an example. If Uncle Raju has diabetes, his son also gets diabetes. That's what we call hereditary. Okay, now colorblindness. Most of humans have this problem, colorblindness. Like a person is colorblind for black to other colors. Like he can't differentiate what is that color like. Let me give you an example. like he can't say, like he can easily see that this is black, but he can't see other colors. Yeah, he can only see black, like some other colors, like yeah. But most common of human beings are facing the problem is greenish red. Yeah, let's see. XCXC means it's a female homozygous dominant for normal color vision. As you guys know, XX is for female, okay? XC, capital C, X small c is female career heterozygous for normal color vision. So it means that the female has no color blindness, that she's fine. Okay. Now this also XC, XC. Both C's are small c's. So female homozygous recessive for red-green color blindness. This, as I mentioned, this is the case, red-green color blindness. Yeah, got it?
My title is Chromosome and Genes. Gene is a basic unit of inheritance. The traits when you inherit from your parents are controlled by genes found on chromosome in nucleus. A human somatic cell contains 23 pairs of chromosome. Okay, this one is chromosome. The chromosome is made up of DNA, deoxyribonucleic acid, and protein. Okay, the DNA is made out of nucleotides. The nucleotides is made up of phosphor groups, deoxyribones, sugars, and nitrogenous base. Okay. Alright, the nitrogenous base have 4 types, which is adenine A, thymine T, cytosine C, guanine G. Okay. A DNA molecule contains 2 polynucleotides strained together to form a double helix. The 2 strands are antiparallel, that is, facing opposite direction. For example, the cytosines link to the guanine and the adenine link to the thymine by hydrogen bonds. The sequence of nitrogenous bases is from the genetic that determine the characteristic of organism. Alright, gene is important. That is determine the characteristic of organism. For example, in humans, there is genes to determine the races, blood type, and they also have the genes to determine the ABO blood type.
Well, hello, G's. Today we are going to learn about the importance of genetics to mankind. There are 2 types of importance to mankind about genetics, which is selective breeding and biotechnology and genetic engineering, which we will go through now. Guys, going to selective breeding. Selective breeding is mainly used for animals and also plants. It's to increase the crop yields, to increase products with higher nutritional value, to have a greater resistance to pathogens, insecticides and pesticides. As shorter maturity time, more offspring can be produced, better adapted to local environmental conditions. So this is what I wrote in the whiteboard. So what they are trying to say, sometimes farmers who need more money need to have easier crop, right? So they use this kind of method, which is selective breeding.
Oh, no.
Okay. We are moving to biotechnology and genetic engineering. The application is DNA fingerprint, which is to identify criminal cases and also for maternity disputes, which is help to identify genetic diseases in also plants and also humans as well, and animals. Next is Human Genome Project, which is a project that determines the sequence of all base pairs found in DNA of human genome, make maps showing the exact locations of genes for major sections of human chromosomes, and produce linkage maps which inherited traits. For example, genetic diseases can be tracked over generations. Potential of stems and disease cell, which is where, for example, people say if we keep a store of stem cells in the stem bank, we are able to find diseases. We are able to find cures for diseases, which is very effective for our body. And also genetic engineering, which include gene therapy, genetically modified organisms, genetically modified food and also medicine, which is production of insulin.